research topics from my lab, but I think they span kind of different areas of human prenatal genetics and aneuploidy, so that hopefully at least it will spur some interest and discussion in these broad aspects. So, I'm curious, so what classes have you already had in human genetics? What have you covered? Aneuploidy or chromosome abnormalities? What's happened so far?
So, I mean, they haven't had a class on abnormality, but they did take concept overview.
Okay, so there's like a foundational genetics class.
There's been an overview of sort of the types of mutations that can occur.
Good, okay.
And where they originate, males and females and so on, but no details about what those abnormalities come about.
Perfect, okay, sounds good. And also I'd like to say, talking for an hour and a half straight is gonna be a lot, so please interrupt with questions, discussion, and I think there will be a lot of opportunity for that, especially next week. My plan is to assign a couple of short readings in preparation for next week, and then I'll be prepared with some more discussion questions as well. Okay, so just a little bit of background on my lab and the reason why this is going to be kind of biased perspective on prenatal genetics is that I'm coming in, I feel like, through a side door. My background is in evolutionary genetics and quantitative genetics. My lab is all dry lab research, computational and statistical research, trying to pull patterns out of genome sequences and learn about how evolution works. Where does our variation come from? How do the forces of evolution filter that variation across the generations to contribute to our genetic diversity? And so some of that work is looking at evolutionary changes that are happening over thousands, tens of thousands, hundreds of thousands of years. And we can use the patterns that exist in our genomes today to study our relationships to one another, as well as to our extinct relatives and living relatives. But we can also apply some of those same principles to understand evolutionary changes that are happening every single generation. And my lab has focused in particular on just the first few days of development, as well as meiosis, happening even before fertilization. And there's a shocking amount of natural selection that's happening just during those first couple of days of development. And I'll give you just a snapshot of some of the evidence that we have for that. Okay. So as I said, I'm structuring this into these kind of three different topics. The first one is just some background on what forms of aneuploidy are common in humans. The word common is kind of a loaded word. We'll get into that debate, maybe especially next class on Wednesday. But we'll also talk about where are these aneuploidies coming from. And then in the second half or 2/3 of the lecture, I'll focus on some of my lab's own work to try to understand what are the factors that influence risk of aneuploid conception or risk of chromosome missegregation after fertilization. And then on Wednesday, the focus will be more on kind of the practical, clinical tests. How do we actually detect aneuploidy clinically? How is it managed clinically? And again, I'm a basic biologist, and so you're hearing this secondhand, but I do interact a lot with people on the industry side as well as on the clinical side. And in some cases, some of the things that we learn about aneuploidy as basic researchers might actually have kind of translational implications. I'm trying to think about that. Okay. So what forms of aneuploidy are common in humans? So it sounds like you've already covered this to some extent. All variation that exists in our genomes that differentiates us from one another ultimately originated from de novo mutation in the formation of sperm or egg, the human germline, right? And there are these fundamental differences about the timing of maternal and paternal meiosis, right? That are contributing, or helping to explain the differences in the number of de novo mutations that are tracing to the egg versus the sperm. So hopefully if you remember back to your previous lectures, something like 75% of de novo point mutations are coming from the paternal germline, are coming from the sperm, right? And the basic explanation for that is that the male meiosis is starting at puberty and then happening continually. So you have more cell divisions that are creating chances for those de novo mutations to occur, right? In contrast to female meiosis, starts in fetal development, but then rests for a long time, and then only kind of reinitiates at ovulation. Okay. This is not the first paper to describe this relationship, but I thought these figures are kind of nice. If you do sequencing of parent-offspring trios, sequence the mom, sequence the dad, sequence the baby, and look for differences in the baby's genome that identify these de novo point mutations, what you see is that there's increase in the number of de novo point mutations on average, with increasing age of the father, as well as with increasing age of the mother. But the slope and just the total magnitude of the y-axis are higher from the paternal side, right? One thing to note is that a lot of these studies, I think possibly all of these studies, have been done looking at living human individuals, right? Sequencing the parents and then sequencing the baby or child, or maybe it's even an adult, right? And looking for these mutations. But we should also keep in mind that it's estimated that in humans, fewer than half of all conceptions actually survive to birth. This is debatable. I've seen papers recently, trying to revise this estimate. There's not a lot of data. This paper from 2002 is still kind of the most highly referenced paper about this topic. But what we do know is that clinical pregnancy loss is very common in humans. Something like 10 to 20% of all clinical pregnancies, clinically recognized pregnancies, end in miscarriage. But it's also suspected that there are many more losses that are happening prior to that stage. It takes even young fertile couples, on average, months to get pregnant. There's some data suggesting that it's actually not failure of fertilization, but very, very early pregnancy losses, often even before implantation, that are the reason why it can take months on average to get pregnant. Yeah.
Has there been any research on, I guess we probably haven't had the technology, but on if these rates are increasing over time in the modern world? Because there's always lots of research about fertility dropping, but is it a failure of fertilization, or is it a failure of the zygote to thrive, as it seems like here?
So I'm not totally dialed into that research area, but I have followed loosely in the popular media. Yeah, go ahead.
Is the rate, you said in humans, but I'm now curious about other mammals.
Oh, yeah, that's a good question too. So, just to finish my thought about your question, I have heard that, for example, sperm counts and like other things have been declining over time. But actually, so we'll get into the reasons for these early pregnancy losses, and I think that the biggest factor is maternal age, and in many industrialized countries, people are waiting until later in life to get pregnant, and so naturally, like the rate of these meiotic origin aneuploidies is going to increase. But it's a good question, and I don't think we have great data. Even, this is a very fuzzy number, and so I don't think we really know how these are changing. But to the question of whether humans are unique,
It's because it's the age than animals.
yeah, that's a great question too. So, and again, I think data are very limited. We know that in like our closest relatives, chimpanzees, gorillas, etc., even rhesus, that chromosome abnormalities are very common as well. So it's probably not a uniquely human phenomenon. In contrast, in mice, for example, they get pregnant every cycle. And aneuploidy rate in live birth is very low. So my hand-wavy explanation is that this must somehow co-evolve with different life history strategies. Like humans have few offspring and make large kind of investment in offspring after birth, in contrast to other species that have many offspring. But I think we don't have very good understanding of like how this maps out across the phylogeny. Okay, so pregnancy loss in general is very common in humans, and it's been known for a long time that the main cause of pregnancy loss are abnormalities in chromosome number. Those include gain or loss of individual chromosomes. We call that aneuploidy. Gain of chromosomes. You're probably familiar with trisomy 21, which is the genetic cause of Down syndrome. Gain of chromosome is called a trisomy. Loss of a chromosome or missing chromosome is called a monosomy. In addition to aneuploidy, you can also have extra or missing copies of the entire genome. Extra copy of the genome we call triploidy. Missing copy of the genome we call haploidy. And you can also have more complex abnormalities. Questions about those kind of definitions? Okay, so you're probably familiar with trisomy 21. Trisomy 21, 18, and 13 are the three autosomal trisomies, autosomal aneuploidies, that are compatible with birth. Most cases of actually all three of these trisomies result in miscarriage, but in, I guess, about 1 in 1,000 births, you have trisomy 21, that rate increases with maternal age. You can see all three of these forms of aneuploidy are increasing in instance with maternal age. For trisomy 13 and 18, most of those babies don't survive to even age 1. Questions about that? In contrast, sex chromosome aneuploidies are relatively more common at birth, and we think that is because of the inbuilt mechanisms of dosage compensation, where you have inactivation of extra copies of X chromosome, right, given in XX or XY individuals. And so in cases of low XXY, that inactivation leads to subtler phenotypes And those aneuploidies, sex chromosome aneuploidies, can be survivable. Okay. So as I've already, I think, kind of implied, the aneuploidies that you see at birth are the ones that are more survivable, right? But if you look earlier and earlier in development, you start to see more and more different forms of aneuploidy. And if you look early enough, like in just embryos a couple of days after fertilization, you see every form of both trisomy and monosomy and combinations of multiple aneuploidies simultaneously affecting a given embryo. So it turns out that the embryo's genome is not even really active until around the 4 to 8 cell stage of development. Embryonic genome activation happens relatively late in humans compared to other species. And so these first few cell divisions are mostly being controlled by maternal RNAs and proteins that are being deposited in the egg. You do have some transcription from the embryonic genome even at the zygote stage, but the major waves of transcription start around the 4 to 8 cell stage in humans. And so almost all of the data that we have about aneuploidies is coming from in vitro fertilization. That's a big caveat to pretty much everything that I'm going to tell you today. There's a big open question, like how much is what we're seeing in IVF an artifact of IVF versus representative of what's happening in natural conception. But at least for aneuploidies of meiotic origin, those that are originating in the egg, we don't think there's any way those could be an artifact of IVF, right? Like it happened in the egg before IVF. Similarly, aneuploidies can originate in the sperm. That must have happened before IVF. So we can learn a lot just by studying IVF embryos and what's happening with them. And what you see in IVF is that only about half of embryos even survive to this day 5 blastocyst stage of development. About half of embryos stop dividing in vitro around day 3, day 4 of development when the embryo's genome turns on. Questions about that? Okay. I'm mostly saving this topic until Wednesday, where we can get into all the interesting controversies about this. But just to say that a lot of the data that we have about aneuploidies comes from a particular genetic test called pre-implantation genetic testing for aneuploidy that is really one of the first applications of genomics in the clinic. It was developed in the early 1990s, and it's gone through kind of several different technical iterations over time, but the basic approach is the same, where in IVF, patients can potentially produce multiple embryos. And then there's the goal of choosing the embryo that's going to be the one that will be most likely to lead to a healthy birth. And so one way of trying to prioritize which embryo is most likely to lead to birth, to implant, etc., is to do genetic testing on the embryos with the idea that chromosome abnormalities, aneuploidies, are the main reason that IVF fails. And so if you can identify embryos that are chromosomally normal, those potentially would have a higher probability of leading to healthy birth. Sounds logical. In practice, it's been extremely controversial. And even today, there's like a huge debate that we can get into more on Wednesday about whether this actually works. But the basic idea is that you can biopsy. They used to do it actually at the cleavage stage. Biopsy a single cell from an embryo at day 3 of development, or biopsy a few cells of embryo at day 5 of development, and then either sequencing or some other genomic assay to try to identify extra or missing copies of chromosomes, and then prioritize the embryos that have no evidence of aneuploidy for transfer. So for sequencing-based approaches, the way that they do it is actually very low coverage sequencing. You don't need to do very much sequencing to identify whole extra or missing copies of chromosomes, and companies try to do this as cheaply as they possibly can. And so just do like 0.01x coverage of the genome, like kind of sampling a read here or there, but you know what to expect, right? And so map the reads back to the genome, correct for technical biases, and you get these types of plots out. And so here, this is kind of an example of an embryo that would be inferred to be chromosomally normal. It has kind of even amount of reads mapping each of the chromosomes, each of the autosomes, and then kind of half the number of reads mapping to X and mapping to Y. So that would be inferred to be a XY, but chromosomally normal embryo. Meanwhile, this embryo, hypothetical embryo, has too many reads mapping to chromosome 13, and so they would infer that to be potentially trisomy of chromosome 13, and that embryo would be deselected and not transferred in IVF. This says, this slide comes from Nanopore. It's actually much more common just to do Illumina MiSeq, like 50 base pair read sequencing. But it's an evolving kind of genetic test, and so all the time people are kind of trying to come up with better and cheaper ways of doing it. Questions about that? Okay, so my PhD thesis, and actually my lab right now, has been trying to repurpose these data to pull out kind of basic information about aneuploidy. So in the clinic, this is done on a kind of patient-by-patient basis, and they use it to inform decisions about which embryo to transfer. But we can combine data from lots and lots of patients, like tens of thousands of patients, hundreds of thousands of embryos, and ask like what chromosome abnormalities are common, what type are rare, how are they associated with parental age, for example. And so these are not our original findings, but I'm just kind of summarizing what you see in IVF embryos. You can distinguish aneuploidies that are originating from different sources. And so one thing that we can see replicates this really well-known finding in the field is that many of the aneuploidies that we observe are coming from the maternal germline, are coming from egg formation, right? Those start at a baseline of less than 20% frequency, but around age 35, you see this kind of dramatic uptick in the frequency of maternal meiotic aneuploidies. In contrast, aneuploidies originating in the paternal germline and sperm formation are very rare. They affect like 1 to 5% of embryos, and there's more recent data showing that they affect about that many sperm as well. And they're not significantly associated with paternal age. Here I'm showing with maternal age, but also there's no detectable statistical association with paternal age. There's a third category of aneuploidies that is very controversial, but is a big focus of my lab's work. Those are aneuploidies that are not originating in the formation of the egg or formation of the sperm, but are actually happening after fertilization during the first few cell divisions of the embryo. You can have chromosomes missegregated. And it seems like actually there are studies recently doing like live imaging of chromosome segregation in embryos. It seems like the first two mitoses in particular are very prone to chromosome missegregation. The reasons for that, we're not totally sure. It might be that cell cycle checkpoints are more permissive in those first couple of divisions. It might have something to do with embryonic genome activation. Maybe some of those checkpoints become more stringent after transcription from the embryo. Those three different sources of aneuploidy have different implications for the embryo. So this is just showing in normal fertilization, you have chromosomally normal sperm and egg, the zygote is normal, and so all the resulting cells theoretically are chromosomally normal. In contrast, in meiotic origin aneuploidy, like an error occurring in an egg, the zygote has the wrong number of chromosomes, and so all of the descendant cells, that aneuploidy propagates to all the descendant cells, clonal division of the embryo. However, in this third category, where you have mitotic error chromosome disintegration after fertilization, you end up with this phenomenon called mosaic aneuploidy, where different cells of embryo have different chromosomal complements. So you probably already covered in your other classes, or this class, somatic mosaicism mutations that can happen during development. This is like a very severe form of somatic mosaicism that's happening early in development, and it's very common. How common? Topic of intense debate, and I have maybe an outlier view on it, but I think every embryo is chromosomally mosaic by the blastocyst stage.
And do those usually come to term, or does that usually result in death, or not sure?
Great question. Yeah, I'll get into it. Yeah. It, short answer, it depends. Okay, so how are those meiotic errors actually happening? If you look at your genetics 101 textbook, it'll talk about non-disjunction, the failure of homologous chromosomes to separate.
Sorry to interrupt, but
Yeah.
you're gonna have to be able to draw it out for the course. Error, it was 1 and 2.
Right, so in meiosis 1, you have separation of homologous chromosomes. In meiosis 2, separation of sister chromosomes, right? And so you can have these non-disjunction events that are occurring during either meiosis 1 or meiosis 2. And if it happens in meiosis 1 versus meiosis 2, the number of kind of products of meiosis affected with aneuploidy will differ. Okay, so here's an example of non-disjunction on meiosis 2, where you have one of these gametes having a trisomy, one of them getting a monosomy.
Most trisomies occur as a result of non-disjunction in meiosis 1.
Yeah, we think most of the meiotic trisomies are happening in meiosis 1, but it varies actually by chromosome, and it also varies with respect to maternal age in different ways. So, and actually, this whole thing is, the more we learn, we found out this is an oversimplification. I'll show you that. So anyway, these, if the egg or the sperm is aneuploid, those aneuploids get transmitted to the zygote. However, just a couple years ago, one of my collaborators in Denmark did genetic analysis, not just of the embryo, but also of the polar bodies in meiosis. And so there you can look at all the products of meiosis, and you can disentangle like which of the aneuploidies are meiosis 1 non-disjunction events versus these two different forms of meiosis II errors. And actually, so the first one is kind of the one that I showed on the previous slide, just the normal meiosis II error, sister chromatid missegregation. But there's actually a second type of error that we didn't know about until just a couple of years ago, called reverse segregation, where you have separation not of homologous chromosomes in meiosis I, but sister chromatids segregate in meiosis I. And then homologous chromosomes segregate in meiosis II. There's a possibility that that can actually happen correctly. And so in the majority of cases, when that happens, you actually get the formation of normal gametes.
So, all the students we failed, they do it wrong.
Yes.
They had a point.
So you can have this reverse segregation phenomenon. Meiosis happening in the opposite order, and it turns out totally fine. But in a fraction of cases, a little less than half of cases, the chromosomes will missegregate in meiosis II. So we can think of these like three different categories of meiotic error: meiosis I non-disjunction versus premature separation of sister chromatids versus reverse segregation. And what they showed in their study, so my collaborator had access to oocytes from adolescent cancer patients. So whereas almost all of our data coming from IVF is from patients of older ages, in this case, they also had access to eggs from teenagers. And what they see is that we've been so focused on these aneuploidies that are increasing with maternal age. Those are mostly these meiosis II errors, this premature separation of sister chromatids and reverse segregation that are increasing with maternal age. In contrast, these meiosis I non-disjunctions are actually elevated in younger patients and decrease with maternal age. So you have what she referred to as a U-shaped curve of aneuploidy. We're typically only focused on this part of the curve, but and it kind of makes sense. We'll get into the actual mechanisms that are holding together the chromosomes, but you essentially have too much chromosome cohesion in these younger age categories, and that's what's leading to these higher rates of meiosis I non-disjunction.
Is reverse segregation a source of homozygote uniparental disomy?
So I think any of these mechanisms can contribute to uniparental disomy. I think most cases of uniparental disomy are thought to be because of a so-called trisomy rescue event, where you have a trisomy in the zygote, but then one of these post-zygotic chromosome missegregation events where a chromosome is lost, and then that rescues a clone of cells within the embryo that can then go and contribute to most of the embryo. So in these uniparental disomy cases, the normal cells kind of start to take over the embryo, and you don't detect a small fraction of trisomic cells. And I think that can originate from any of those three mechanisms. Okay, so here's some more direct evidence of the point that I made earlier. So we see in embryos that aneuploidies of paternal origin paternal meiotic origin are very rare, but just in the last couple of years, it's actually been possible to do single gamete sequencing, single sperm sequencing. This is a paper from Steve McCarroll's lab, where they did that on a few thousand single sperm cells. And what they see is that about 1 to 5% of the sperm cells have chromosome abnormalities. Relatively more losses than gains, more monosomies than trisomies originating from the paternal germline. Okay.
So one question that Dr. Ravi asked once was, I don't know, but it's like a question that, why would the evolution design the system where single mutation come from the dad and the big mutations come from, the big chromosome mutation come from the mom? There was no answer. It was just
Yeah, that is a great question. Why? Yeah.
It could be random, and then we stuck with it, but.
What were your thoughts about that?
It could also just be a cause of the mechanisms, right, of reproduction. So
Oh yeah, it is definitely a cause. I mean, the fact that the mom is, there are causes, but usually if it's not useful, it gets rid of it. The fact that it's sort of, I don't know, maybe it is random that it's happening this way. I mean, we know why it's happening, but why is it happening this way?
Yeah, so I'm coming from more evolutionary genetics background, so that question fascinates me, and like ultimately, it's like one of the drivers of my research program, but it's so hard to answer. And so I'm interested in like what are the possible trade-offs, fitness trade-offs that could like explain why something that seems like so harmful is still like able to persist. And I'll talk about it a little bit in this work, but one idea is, again, very hand-wavy, is that one of the risk factors for aneuploidy is abnormality in crossover recombination. These crossovers that are kind of shuffling the genome are also essential for accurate chromosome segregation. But they're also mutagenic. They induce point mutations and structural variation. And so it feels like there's the potential for a lot of trade-offs built in there. Like you need it to play these multiple roles for one thing, but also it has like a harmful like fitness cost associated with it. And so there must be some like really interesting trade-offs going on. Plus, there's potential for alleles that kind of cheat meiosis, like meiotic drive, where especially in the female germline, only one of the gamete, or only one of the products of meiosis becomes the actual egg. The rest of them become port bodies. And so if you're an allele, you want to be the one that gets into the egg, right? And so if you can cheat the process of meiosis, and there are examples of this in model organisms, to do that, then you'll spread through the population and fix in the population. So there's this constant like arms race that's going on within our genomes, and my lab's interested in finding evidence of that in humans as well.
And this is also true in other species. It's mom has the big mistakes and daddy has, I mean, both are big in terms of consequences, but like big chromosomal mutation in mom, and the dad is like point mutation.
That's a good question. Yeah, I again don't know like how far out this in phylogenetic space this has been looked at. Yeah, certainly for our relatives, yes.
But there has to be sexual reproduction.
Yeah, and it's only whole chromosome aneuploidies, like large like structural variation, things like that, are also mostly coming from the paternal germline thing. But it's an interesting observation for sure. So actually getting to exactly that, we already mentioned that female meiosis actually starts during fetal development, right? You have the pairing of chromosomes and the formation of these crossovers, the exchange of material. But then meiosis pauses prenatally and doesn't restart until ovulation, decades later. And so the chromosomes have to stay in this configuration, in this like paired crossed over configuration for decades. And the crossovers themselves are actually important for establishing the chromosome pairing, the pairing of homologous chromosomes, as well as kind of maintaining cohesion of the chromosomes during meiotic arrest. In addition to the crossovers, you have the formation of this temporary kind of zipper-like structure of proteins that forms in between the homologous chromosomes called the synaptonemal complex. And then you also have the loading of these protein rings called cohesins that hold together the sister chromatids. And those also have to be maintained over this decades-long period of meiotic arrest. And if you have deterioration of this chromosome cohesion, that's contributing to this premature separation of sister chromatids or to this reverse segregation phenomenon, which increase with maternal age. So that's kind of the like very basic explanation for the maternal age effect on aneuploidy, but the details are still very unknown, and that's a very hot topic of research. For example, Melina Schu's lab in Germany is like using mouse models and pigs as a model for studying meiosis and mapping out all of these different proteins that are involved in these processes. Yeah.
Are cohesins stable proteins, or what's their half-life? Like basically, is there the one you have like long,
Yeah.
less long, or?
So you're going to stretch the limits of my knowledge, but these cohesins are only loaded during fetal development, so they're not turning over. Yeah, yeah.
So what you got is what you got in the beginning?
That's right, yeah. And they deteriorate over time. And so there are some of this, so it's a multi protein complex that forms this ring. And some of the components of that ring are meiosis specific, and some of them are shared between meiosis and mitosis, which is also interesting.
So maybe there's
Yeah.
some variance that makes women more fertile longer.
I'll tell you one story about that. Yeah. That's actually my last
sorry,
area of
I swear I
research.
didn't.
No. That's a great.
I didn't wanna steal your
No, you set it up perfectly. So to kind of study these processes, again, like one very useful source of data is data from in vitro fertilization. And so we have a collaboration with a genetic testing company in California called Natera that is one of the companies that does this prenatal genetic testing. And so we can retrospectively reanalyze these de-identified data from embryo biopsies, about 145,000 embryo biopsies. And Natera is actually kind of unique, unlike the example I gave you before about DNA sequencing for pre-implantation genetic testing. Natera actually does single nucleotide polymorphism array genotyping of the embryo biopsies, as well as the parents, the patient and partner. The reason that they get data from the patient and partner is that it's only a few cells that are coming from the biopsy, like 5 to 10 cells, so it's a low amount of input DNA. It used to just be one cell that they would take. And so the data that you get from the array is very noisy. And so Natera's whole kind of technical innovation is that all of the chromosomes that you could possibly see in the embryo must have come from one of the parents. And so you can kind of clean up the noisy embryo data using your knowledge of the parents' genotypes, right? So that's the basic approach. They do this SNP array genotyping on parents and embryos and use that to infer aneuploidies. But as a human geneticist, I get very excited about this because unlike living human families where you can have maybe a few offspring, and you've already learned about like how useful it is to have pedigrees in human genetics, right?
They have.
Here, you can have 20, even like 30 embryos from the same set of parents. And so that's a lot of replications of meiosis that we can use to study basic aspects of genetic transmission. Okay, so this is where it gets to be a very biased view of this topic, because I'm going to talk about my lab's own research. This is all unpublished work that I'm going to be telling you about. And my postdoc, Arjun Vedanta, developed a method to analyze these data. And so one kind of cool thing, when you have multiple embryos from a given set of parents, it allows you to phase the genotypes of the entire chromosome, right? You can use this kind of majority vote approach, and so for all of the heterozygous mutations, you can assign it to one haplotype or the other. So we can reconstruct the maternal and paternal haplotypes.
So these sequences are long enough that you can put it in the.
So there's no sequencing.
It's SNP.
It's all SNP array genotyping, and so the basic strategy is to choose the phase that minimizes the number of crossovers that you have to invoke across the embryos. So we kind of phase by parsimony.
Mm-hmm.
There will be some errors, but as you have more and more embryos, the number of errors declines. So if you have at least 3, you can do this kind of majority vote approach, and it allows you to get this kind of long-range phasing. And there are other ways of phasing. Maybe you've already learned about them. Other approaches to phasing, for example, use long-read sequencing, or you try to get lucky enough to catch multiple heterozygous mutations on the same sequencing read. That allows you to know that, say you have these 2 heterozygous variants, it lets you know that like these 2 alleles go on this haplotype and these 2 alleles go on this haplotype, right? But that only works if you get really lucky. Like humans are not that genetically diverse. We only have a heterozygous SNP, one in about 1,000 base pairs in our genome. And so if you have paired end Illumina reads, you have to get lucky enough to like land one end on there, one end on there. Yeah.
I mean, if you were doing this using long-read sequencing, are there also like uniquely parental like methylation patterns that you could also look at?
Yeah, that's another strategy people use. So, for example, Nanopore can directly read off methylation patterns.
The company's not going to invest in sequencing.
That's the thing, yeah. So a lot of my lab's work has been trying to squeeze out information out of like very sparse data, because companies are trying to do this as cheaply as possible. And to detect aneuploidies, you need very little data. But as the field keeps moving, I do expect that eventually, like a lot of genetic tests, and we'll talk about this on Wednesday, a lot of genetic tests are just going to get merged into one assay, genome sequencing. And so there will be much more data to work with, and you can use these alternative strategies potentially. Okay, but for now we have to work with this. And so we take the embryo data then, and then we can trace through the parental haplotypes and identify the path through the parental haplotypes that best explains the observed data from the embryos. And so, for example, here you have this paternal chromosome in the embryo that the first half is coming from this paternal homologue, the second half you have to invoke a crossover, you switch to that paternal homolog. These switches are the locations of inferred crossover recombination events. Here I'm illustrating that sometimes you actually have to invoke 3 paths through the haplotypes rather than just, one maternal, one paternal. Sometimes you have evidence of multiple paths. And so here's an example of a trisomy. Yeah.
So, like, being a hidden Markov model, is it really kind of just as simple as saying, like, okay, given this SNP state here, what is the most likely, like, crossover event that would lead this SNP to be this SNP, and then that one to be on this one?
Yeah.
Like, is it kind of just walking through and...
That's exactly how it's working. And so we have these probabilities of transitioning from like one haplotype to another that are just based on kind of average recombination rate across the genome. But you have a much higher probability of staying in your given kind of haplotype state. So hidden Markov model is very common, powerful statistical approach for working with kind of data where the order matters and trying to infer the most likely path through these different haplotype sequences. Okay, so what we actually see in these embryos, most of what I'm showing you is not like an original finding from my lab. I'm just showing it to you with respect to our data. But here's one result that we replicate from the field, where if you look at these blastocyst stage embryos, these are embryos at day 5 of development. There's ascertainment bias here. I should note, only embryos that actually survive to day 5 of development and look good morphologically get tested. So I'll come back to that later. But of these embryos, about 2/3 look chromosomally normal based on their biopsies. 22% have aneuploidies of single chromosome. But you still see, even in these like surviving good morphology embryos, 12% have aneuploidies affecting multiple chromosomes simultaneously. And actually these we don't even call aneuploidies, we call these whole genome abnormalities, right? For example, triploidy, 3 copies of every chromosome. The number of aneuploidies varies across chromosomes. And so you can see chromosome 15, 16, 21, and 22 have much higher incidence of aneuploidy than the other chromosomes. And again, most of these aneuploidies are coming from the maternal germline. We can infer that because we know which of the chromosomes came from mom, which came from dad. And it's these maternal putative meiotic aneuploidies that are the ones that are driving this kind of chromosome specificity, right? It looks like chromosome 15, 16, 21, and 22 are more susceptible to maternal meiotic errors for some reason. You might say that, well, chromosome 21, you already know that that's like one of the survivable trisomies, so maybe this is like a selection bias. This embryo is surviving better to day 5 blastocyst stage. That is true, but actually even if we look in eggs, you still see that chromosome 15, 16, 21, and 22 are the ones that are particularly susceptible. And by the way, we have no idea why. That is a totally open question as far as I know. These are smaller chromosomes. Maybe it has something to do with the actual structure of the chromosomes. These are aprocentric chromosomes. They have very small, short arms. Yeah.
Is it related at all to gene density?
Only in the sense that these are small chromosomes, yes. But
Not the number, but the density.
yeah. So one thing, I don't have a slide to show it, but there's already been quite a bit of selection prior to the blastocyst stage. And so we see that by the blastocyst stage, trisomies are more common than monosomies, for example. And if you compare the ratio of monosomies and trisomies, you see that, in particular, larger chromosomes with more genes have, sorry, I'm like mixing it up in my head now, but the monosomy-trisomy ratio varies by chromosome. And so it looks like the ratio is about the same, monosomies to trisomies for these short chromosomes. But monosomies of these larger chromosomes are particularly harmful. Yeah.
So I know that when we put cells in culture, even if they're primary cells, then if you grow them long enough, they'll get chromosomal abnormalities of some type, even if they're not cancer. Is this sort of pattern true for cells in culture, that of all the abnormalities that you can get in a culture, cell 15 is always 16 is going to be higher than 11?
I'm not sure for mitotic chromosome
Just
segregation.
in the sense that maybe it would be like a clue either for selection or mechanism. I don't know.
That's interesting. So at least in embryos, what we see is that the post-zygotic errors, the mitotic errors, don't have
This
any
pattern.
like this. They seem to be more consistent across chromosomes. And they also obviously don't care, well, maybe it's not
Yeah.
obvious, they don't care maternal or paternal homolog. Mitotically, chromosomes wouldn't segregate kind of
So
randomly.
it seems like it's more meiotic specific.
Yeah, it has something to do with the actual like mutation generating mechanism. One hypothesis, maybe it has something to do with centromere variation. You probably learned from your classes that only like in the last couple years have we gotten good methods to look at centromeres. And so that's an area for, you know, research in the coming years, like maybe something to do with those haplotypes is explaining some of these differences across chromosomes. We can fit statistical models to this maternal age curve, and one nerdy but cool to me statistical observation is that this is well fit by a quadratic model. Just logistic regression with a quadratic term, which implies that the rate of maternal meiotic aneuploidy accelerates at a constant rate with age. So it doesn't, it's not a line, but a quadratic, it has this curve to it, which means that it's accelerating at a constant rate. Oops, oh no, wrong button. Meanwhile, even in this like very large sample, we see no paternal age association. Again, we can map out those meiotic crossovers and assign those to maternal versus paternal origin. And this is also replicating a finding from the field that there are more maternal crossovers than there are paternal crossovers. We actually don't see it in our study, but in the literature, there's an observation that the rate of crossovers, maternal crossovers, increases with maternal age, but only a little bit. And it's an open question of like, is it actually that crossovers are increasing, or is it driven by selection against aneuploid embryos, explaining this change in rate of crossovers with age? Because we have the parental genotypes, we can try to identify genetic variants in the parents' genomes that are associated with either crossovers or with aneuploidy. And again, we think that crossovers themselves are one of the risk factors. Abnormal number or location of crossovers are one of the risk factors for the formation of aneuploidy, so we want to study both of those together. So we can scan through the maternal genome and the paternal genome, look SNP by SNP, and ask, carry this particular allele of this SNP, do you have higher or lower number of crossovers in your embryos? And so when we do that, we replicate some of these really well-known findings from the field. We identify genetic variants in meiotic crossover-related genes. This is a gene RNF212 that's involved in crossover regulation. C14orf39 is actually part of the synaptonemal complex that I showed you before, that protein structure holding together the homologous chromosomes, as well as common inversion polymorphisms. So it's known that large inversions can suppress recombination in heterozygotes within the inversion region, and then actually increase crossovers outside of the inversion region. So common genetic variation is contributing to variation in the number of crossovers. Some of those risk or variants that are controlling crossovers are shared between the sexes, but many of them are sort of sex specific. Okay, so this was number of crossovers, but also we can look at crossover location. We can define that in a couple different ways, like asking on average how close is a crossover to the centromere, how close is it to the telomere, and we can identify again, these are, this is actually part of the cohesin complex, but we can identify these genetic variants that are in kind of key meiotic machinery that are associated with like where exactly the crossovers are placed in the genome. And then you might have already touched on this in your classes, but crossovers are not happening randomly in the genome in humans and other vertebrate species. There's massive heterogeneity in crossovers. There are some parts of the genome that have like 1,000, 10,000-fold increased rate of crossovers. We call those recombination hotspots. And something like, I want to say like 20% of all crossovers are happening in those like very short recombination hotspots. When we look for genetic variants associated with recombination happening inside versus outside of hotspots, we pull out this master regulator of recombination hotspot determination, PRDM9. PRDM9 is a transcription factor that is binding to a very specific motif in the genome that recruits, it binds, then it recruits the SPO11 protein that makes the double-strand breaks that initiate crossovers. Okay, so all that work was focused on crossovers, but it's largely replicating reports from the literature, and one of the most important papers was this paper from 2019 that I think I signed somebody to present on Friday. That's a study by Decode Genetics. It's a company in Iceland that, so Iceland has these meticulous records of genealogies, as well as like a very high rate of participation in genetic research. And so this is a study from this company where they did sequencing of parent offspring trios and mapped out crossovers and also looked at de novo mutations. And they found that crossovers are themselves mutagenic. They can induce de novo mutations near the location of crossover double-strand breaks. But that was all done on living individuals, right? And so they weren't able to look at chromosome abnormalities and the potential shared basis of crossovers and aneuploidy. But they did note that one of the variants that they found associated with crossovers was in this gene SMC1B, which encodes a cohesin, and it is associated with 3 different crossover location phenotypes. And they said whether this variant or others identified in our study is associated with greater risk for aneuploidy remains to be seen. And why is that? Why would they even think that? Again, I mentioned that crossovers, the location of crossovers, the number of crossovers is thought to be important for aneuploidy risk. Crossovers are important for pairing of chromosomes and holding the chromosomes together during meiotic arrest. And so in our study, we looked at the number of crossovers on normal disomic chromosomes in euploid embryos, chromosomally normal embryos, versus aneuploid embryos. And what we see is that even on the normal disomic chromosomes, 2 copies, even on those chromosomes, you see that aneuploid embryos have significantly fewer crossovers than euploid embryos. The reason we looked at disomic chromosomes is because when you're looking at the trisomic chromosomes, we think you can miss some crossovers. If you have if you have a trisomic chromosome that looks like this, right? Say that these are the 2 maternal homologs. And a crossover happened right here, and you happen to get both of the reciprocal products of meiosis. These are invisible to us.
It goes back to,
Yeah, we're tracing through the parental homologs, and so we're not going to be able to find these where the crossover is like perfectly aligned, right? Because we missed these, we're focusing only on the chromosomally normal chromosomes and relying on the fact that it's already known in the literature that patterns of crossovers co-vary across the chromosomes in meiosis. So this, sorry, a long-winded explanation, but this is basically a trick. The focus on disomic chromosomes is a trick to get around
so
this
if,
problem.
the aneuploid, it was for chromosome 22. We looked at all the other ones
That's right.
except 22.
That's right, yeah. Does that make sense? Yeah.
Is there any evidence that like too many crossover, like extra crossover elements
Is also associated with
Yeah.
aneuploidy. So I think this is a kind of average observation. We see that too few crossovers is associated with aneuploidy, but I have some results. I don't know if I have a slide, but I think this is a big oversimplification. What we find is that genetic variants associated with crossovers don't have a consistent directional effect on aneuploidy. We see that they often also influence aneuploidy risk, but not in a consistent direction, which implies to me that like it's very kind of variant specific. It's a good question. Sorry, I jumped ahead a little bit, but we're also interested in doing the same thing for aneuploidy. Can we do a genome-wide association study for aneuploidy and find risk factors in the maternal genome that may predispose a patient to make more aneuploid embryos or less? And we mapped 2 genome-wide significant associations. The first one's on chromosome 6. The way we do this is we split the sample. We hold out part of the sample as like a replication set and use the rest as a discovery set. And so when we reanalyze that held out sample, this one does not replicate in our held out sample, so we're less confident in that hit. This one does replicate in the held out sample, and so I'll focus on that one. It's on chromosome 21. It's kind of circular, but it's a common allele. GWAS is powered. Genome-wide association studies are well powered to identify common variants associated with phenotypes.
So you
right?
did GWAS on your single embryos?
Not on the embryos, but on the patients. So
Oh, the mom and dad.
the phenotype.
Oh, okay.
The phenotype is aneuploidy in the embryos. The genotype is coming from the parent's genome, and so this is showing GWAS on the mom's genome for variants that are associated with aneuploidy in her embryos. Does that make sense? Same thing with the recombination one. We're getting the phenotype from the embryos, but the genotypes are coming from the patients. And okay, here's our hit. It's a common allele. It's slightly less It's less common in Africa, more common in non-African populations. It's a small effect, so the more you learn about GWAS, this is basically what GWAS can find. That it can find common variants with small effects, right? And so this variant has about every copy of the risk allele increases the rate of aneuploidy in embryos by like 1.5%. The variant, so variants don't travel alone, they occur on haplotypes, so you get these like peaks of association, so you have to kind of zoom in to see like what genes does this span. It spans a couple of genes, but one is a very obvious candidate. That's that same gene, SMC1 beta. It's part of the meiotic cohesin complex. It's part of this ring structure that's holding together the sister chromatids during that long stage of meiotic arrest. And so it was known since mid-2000s. This is work from Pat Hunt's lab at Washington State University. Knocking this gene out in mice leads to defects in recombination and chromosome pairing, and ultimately leads to aneuploidy in the formation of eggs. Because we also have the phenotype data, GWAS data from the crossovers, we can go back to that, and we see that the same variant that's associated with risk of aneuploidy is also associated with number of crossovers. Fewer crossovers per embryo, crossovers happening further from centromeres and telomeres, so more toward the center of chromosome arms. I won't go into detail about this, but there are other types of prenatal genetic testing data. There's data from clinical miscarriages, and we see that even at that later stage of development, the same variant is associated with higher risk of aneuploid miscarriages. And we can mine the literature from association studies of timing of these reproductive events in women, age of menarche, so first period, and then age at menopause. The same variant is associated with later age of menarche and earlier age at menopause, so like a shortened kind of reproductive window. So we don't actually know how this is working. It implies that there must be some type of signaling from the ovarian reserves that kind of impact the timing of these different reproductive aging events.
It's like you have fewer good eggs, start later.
Yeah, something like that, yeah.
Could it also be from the placenta? Like, if there was, like a puncture in the placenta could lead to miscarriage? Not that that is a common thing that happens, but just damage to the placenta in general.
I think so, yeah. I'll have to think more about how that could play a role.
Maybe for the early menopause, if you had a child, then the placenta could have done some signaling that stays on for some reason.
That's interesting,
But
yeah.
for the menarche, no. But for the menopause, if they, basically you could maybe split it between people that have had babies and those people.
Oh, that's a good idea, yeah. We have,
Although
unfortunately,
maybe that alone may influence menopause.
we don't have a ton of data because this comes from a genetic testing company rather than a fertility clinic. Our data on kind of patient clinical history is pretty limited, but I do like that idea of kind of looking at past pregnancies as one kind of covariant. One big goal in human genetics and association studies is try to understand the actual mechanism by which the causal variant is driving associations. And so one way to get at that is by looking at gene expression, effects on gene expression. And in this case, we're going to an external data set called GTEx. you might be familiar with. It's RNA sequencing of tissues from post-mortem donors, where they've also done genome sequencing of the same individuals. So they know genetic variants that are associated with expression of genes in different tissues. We call those EQTLs, expression quantitative trait loci. And so the aneuploidy risk allele in this case is actually associated with the expression of that same gene, SMC1B, across many different tissues. Unfortunately, we don't have data from fetal oocytes. That's like probably the most relevant tissue. But what we know is that genetic effects on expression tend to be correlated across tissues. And what you find in EQTL studies tend to be things, actually, by definition, what they're looking for are variants close to the gene. And what you tend to find are like large effect variants in the promoter that are associated with the expression of the gene. And those, it kind of makes sense, like it doesn't matter what tissue, what cell type, if you screw up the promoter, it kind of has consistent effects across different tissues. So again, like one challenge in association studies is that you don't just pull out a single variant, like you get a whole haplotype. And we want to know like which of those variants is actually causing the phenotype. And that can be really hard to figure out, right? These are all in linkage disequilibrium with one another, like they're hard to disentangle from one another. So one way to address that is to get a more genetically diverse sample. In more genetically diverse samples, there's been more recombination in the history of the sample, and so those haplotypes get narrower and narrower. Meanwhile, we think genetics works the same in different the more we learn about kind of human genetic diversity, we think EQTLs are actually very consistent in their effects across different populations. There is not a lot of evidence of like genetic interactions or epistasis. If you have a variant, conditional on having a variant, it will have a very consistent effect, regardless of genetic background. Of course, there are going to be exceptions to that, but in general, kind of, that's a basic observation from our field.
Do we have enough observations?
So my lab's been trying to address that. we did RNA, not for this study, for a previous study. We did RNA sequencing of cell lines from people from the 1000 Genomes Project. Caveat is that this is just one cell type. Immortalized cell lines from blood immune cells, lymphoblastoid cell lines, but spread across these 26 different populations in the 1000 Genomes Project. And if you look even in that cell type, you see that that variant is an EQTL of SMC1 beta. But unlike GTEx, which is mostly European individuals, and you have those long haplotypes, here we have representation from other populations, so we can start to narrow the peak. And we use an approach called fine mapping to like try to narrow the peak as much as we possibly can and actually figure out, is it really one peak or is it two peaks like right on top of each other? What we find is that it's actually two peaks. One of those peaks we can narrow down just to a single variant. We call these independent signals credible sets. Software tries to narrow the credible set as much as it possibly can. Sometimes you can't narrow it down to just a single variant. You still have, even in this diverse sample, too much linkage disequilibrium, so you can't disentangle. But for one of those variants, we can narrow it down just to a single variant right in the promoter of SMC1B. For the other one, we're still left with about 8 variants. But that was kind of an interesting observation because it looked, if you only looked at GTEx, it looked like it was probably just a single variant driving the effect. But in the more diverse populations, you're actually able to disentangle this phenomenon called allelic heterogeneity, where it's multiple variants like traveling together in European populations, but all of a sudden when you look in more diverse populations, you find out, oh, actually 2 different variants going in the same direction.
I think some variation though is not gonna, you said, you're gonna find EQTLs in the promoter. and it will be true in every cell type, but there is also a lot of other stuff that is very much cell-specific that you
Yeah.
cannot find if you don't look at the right cell type.
That's right, yeah, these enhancer variants are gonna be more context
But
specific.
this one, yeah.
And,
I just wanna tell them so they don't.
absolutely, yeah, yeah. And, trans EQTLs, so, there are a lot of technical limitations, so, EQTL mapping has been done, in cis, like looking just around a window around a gene, largely for practical reasons. Like we're only well powered to identify variants, because if you think about it, okay, sorry, I'm going, this is the advanced human genetics class, so we don't have to stay on anybody. But if you think about it, there's like, I don't know, millions of SNPs in the genome, 3 million or so heterozygous SNPs per individual's genome, but say we assay like 1 million of those, right? And then in EQTL studies, you're associating these SNPs with the expression of every gene, right? And there's like 20,000 protein coding genes in the genome. Multiply the two big numbers together is a huge number. And that's a problem for two reasons. One is like purely like computing reason. Just like writing a for loop and doing that many tests will crash your computer, even if you have a fancy computer. The second is this p-value threshold, right? Like, I'm sure you've learned like p less than 0.05 is kind of like the standard, we call that statistically significant. But if you do multiple independent tests, you have to lower your threshold farther and farther to control the risk of false positives, right? And so one way we can address that is using Monferroni threshold, where you take that 0.05 and divide by the number of independent statistical tests. Divide 0.05 by this number, and it's going to be a p-value that is like so tiny you'll never be able to detect anything. Does that make sense? And so for that reason, we don't do the EQTL mapping for the whole genome. We just look in a little window around the gene. But it turns out that you're missing most of the stuff if you do that. 80%, it's estimated something like 80% of the heritability of gene expression is explained by variants in trans, not in cis. And so these cis genetic effects tend to be bigger. They tend to be concentrated in the promoter, et cetera, or nearby enhancers. But most of the heritability of gene expression is happening outside. And those other variants are probably more context specific. Okay.
And I think there was a journal club that we did where they showed, it was like a whole study about all the EQTLs that you cannot find in T-tags because of all
Yeah.
these diseases, that you find them in UAS.
Right. Yeah, and all of this, including our work, is only focused on the common variation. Meanwhile, in humans, our populations have grown very fast, very recently in evolutionary time. And so that is casting a big net for new mutations that haven't had a chance to increase in frequency yet. And for that reason, a lot of traits, especially for traits that are tied to fitness like this, we think this is tied to survival, right? Their variation in those traits should have kind of a larger, rare variation should play a larger role in traits like that. Okay. Just in the the last couple minutes. There's a kind of complementary approach that we applied in our study, but it's a general approach you might be interested in for human genetics. Rather than doing this association where you're associating genetic variants in the genome with the phenotype, we can use this approach where we take eQTL mapping data, like the GTEx data that I told you about before, to take the genotypes and then try to predict the expression of the genes, right? If we know from some external data the relationship between genetic variation and expression, we can actually try to predict expression of each of the genes in any given tissue, right? And so we did that. We take our data from the Natera patients, from the IVF patients, and we predict gene expression in all their different tissues. And then rather than associating the genetic variants with the phenotype, we associate the predicted gene expression with the phenotype. We call that a transcriptome-wide association study, or TWAS. And so here, you can see that we pull out again SMC1B, as well as a few other genes in the region that we think are kind of co-regulated by the same haplotype. So again, we suspect that this is driven by linkage disequilibrium and haplotype effects. But we also pull out some additional variants like this predicted expression of C14 or 39, that synaptonemal complex gene, is associated with aneuploidy in embryos, as well as this gene NCAPD2, part of the condensin complex, involved in chromosome condensation during meiotic prophase 1. So this is kind of an alternative approach here. Each little dot is not a SNP, but it's an individual gene, predicted expression of an individual gene.
So you predict the expression of these genes in the based on the GTEx data.
So we predict the expression in a given tissue because GTEx is tissue data.
Right, but then what value do you actually use for your?
So we were combining it combining the signal across all the tissues.
Okay, so
Yeah.
more than enough tissue.
Yeah. So GTEx is 50 different tissues, so we actually do 50 TWASs. And
that
combine
value
all the
presumably
results.
the-
Unfortunately, none of those are for fetal oocyte, but again, we're using this logic that hopefully these other tissues are providing some proxy. Yeah. Okay, I think
It's good.
we'll stop there for today.
He wasn't so bad in
I didn't even talk about mitotic errors, so we'll talk about mitotic errors next week, but I'll also send a couple. Don't be intimidated by this. It's like popular media articles that have to do with, in addition to kind of the technical aspects of prenatal testing, some of the ethical implications of prenatal testing, where this is going. So, cool.
Thank you.
Thank you.